If you're the lucky person to teach the unit on animals, we have a lot of fun things in store. And you should have at least one live animal in your classroom. But before you run out and decide just what type of animal to get, you should look at all the different ramifications. For example, does the animal need any special care? And what are you going to do when vacation time rolls around? Well, the best thing is to check with the local pet store and learn a little bit about the animals before you decide to get one. Mammals require the most care, whether it's a hamster or a rabbit. And you should also consider whether the animal is going to be sleeping all day while the students are observing it. This little rabbit started out as a pet in my classroom, and I couldn't resist his cute little face. So I ended up taking him home for vacation, and he's lived in my backyard ever since. He only has one deep hole, and he still continues to terrorize all the neighborhood cats. Then again, you can always have low-maintenance pets, like these zebra finches. The male is the one with the orange cheeks. If you have fish in your classroom, be sure that the heating element has a plastic cap on the top. This will keep the students from turning up the temperature on your fish. In your unit on animals, you should review some of the primary concepts first. The idea that we classify animals according to their characteristics. Remember that memory aid and that animal classification scheme. King Paul cried out for good soup. You might start taking a look at that. We'll continue to investigate animal adaptations. We'll contrast the difference between cold-blooded and warm-blooded animals. We'll learn how animals reproduce. And we'll see how animals go through different stages in their life cycle. Let's start first by looking at animal adaptations. By looking at characteristics of animals, we're also learning about how they adapt themselves to the environment. So adaptations then are just special characteristics that animals have to survive. Let's take this fish for an example. It is able to live underwater. It does so with the aid of an organ called a gill. How does this gill work? Well, water has oxygen dissolved into it. It has the gas mixed in with the liquid water molecules. When the fish passes water past the gill is able to remove that gas from the water and take it into the bloodstream because the blood vessels are so very close to the flow of water, only about 2 cells thick. When the water gets depleted with oxygen, for example, if you don't put an air stone in your tank, then this goldfish is also adapted to be able to swim to the surface and gulp air. Now how it does this, I'm not quite sure, but it's able to survive even in water that has a low oxygen content. So when you have a fish tank, it's always important to have an air stone. If we look at an amphibian, we see an entirely different adaptation. If we take a look at this frog, we see that not only is it adapted to live in the grasslands because it has nice camouflage on its skin, but it can also breathe in the water or in the air. It can take oxygen directly through its skin because the skin can absorb the oxygen like a gill does on a fish. And it can also breathe with lungs. The number of different types of animal adaptations are much too numerous to list here. A good way to learn about them is to assign an animal to each student and have them report back with the specific adaptation that that animal has. You might have them pick names from a hat. Some good examples might be an elephant, a camel, or a giraffe. Now let's continue with adaptations and look at camouflage in particular and learn a little bit about natural selection. Here we're going to use some different cutout dots from a paper punch. I've got a cupful of green ones and a cupful of yellow ones. We'll take 25 of each and sprinkle them on this piece of green paper. Make sure that none of them touch each other so they're all spread out evenly. Your students can work in groups of 2. Take another paper cup and punch a tiny pinhole on the bottom end of it. The student that's going to pick up the dots looks through the tiny little pinhole and has only 30 seconds to start picking up dots. Can only pick up one at a time and put it in the little dish. After 30 seconds, the other student that's running the clock says stop, and they look to see how many dots they have left. Follow standard scientific procedures here. Make sure your students do more than one trial and average their results if possible. We're going to see this as a predator-prey relationship. Whenever we take a dot off, we've eaten it. The ones we don't get are the ones that survive. At the end of a trial, let's assume that all the dots that are left on the paper can now reproduce. In other words, they double in size. Which ones will have a tendency to become more dominant, the yellow ones or the green ones? And at the end of that trial, let them reproduce again. Eventually your students will realize that all the yellow ones will disappear and the green ones will take their place. This is called natural selection, or sometimes survival of the fittest, and it happens every day. The animals that have the camouflage that blend in good with the environment survive, and those that stick out get consumed by the predators. Next, let's move to another activity, and one you must do. It's not only very exciting and lots of fun, but it's something the students will remember for a long time, and this is the live frog lab. We'll look at how the frog is adapted to the environment. We'll learn about the difference between cold-blooded and warm-blooded animals, and we'll understand more about some of the systems, like the respiratory system inside the frog. I recommend you do this as a demonstration, because if you have frogs for every student or groups of students, you're going to have a real wild time, I'll guarantee you that. You can have a lot of students participate by coming up and making some of the measurements. We have one live grass frog in this tank. Notice that I have it filled with gravel, and I have a double plastic container inside so that I can very easily change the water. The frog doesn't need much to survive. They eat crickets or mealworms and can live on 5 to 10 per week. Make sure that you always have fresh water in there because the frog needs to remain moist. If you can't find a frog locally, you can always order one. They're called the grass frog or Rana pippins. That's the genus and species name, and they're only a few dollars. And by all means, don't be afraid to do this lab, even if you've never touched a frog before. They're a little slimy and quite squirrely, but you'll have a lot of fun and learn a lot by doing it. The first thing we're going to do is make some simple observations about the frog. How long is it from head to tail, not counting the legs? And how does it sit just before it's ready to leap? Then we'll see how far it can jump. We'll put a chalk mark X on the floor, set the frog in it, and measure the distance it jumps for about 5 trials. Make sure that you have some students sitting in the perimeter who can catch the frog as it starts leaping toward them. Then we'll see how the frog swims. And if I can get this frog out of here and put him in the other tank nearby, we'll see how he swims. You see that I have just as much difficulty as you might have. Slimy little frog. There we go. Oops. We could spend the whole time. It's okay to grab him by the legs too. Okay, we put him in the tank here, and then we can start to see how this little frog swims. We see the classic frog kick, and as you look closely at the frog's feet, you'll notice another adaptation, webbed feet, to enable the frog to swim better. Man has learned much by studying different animal adaptations. We sometimes swim with the frog kick, and of course swim fins can be a real help. Now let's do an experiment to investigate how the water temperature affects the respiration or breathing rate of the frog. If we look back at when the frog was in the tank, we see that it breathes in a kind of unique way. First it takes air in through its nostrils. Then it closes the nostril and gulps the air into the lungs with his throat. You can measure the breathing rate by counting the number of nostril movements per minute, or the number of throat gulps per minute. You should discover that the throat gulp number may be 4 to 5 times the nostril movement number. When the frog is floating in the water, students should notice that the nostrils, the eyes, and the ear membranes, which are just behind the eyes, are just at or above the water line. Here, the frog is breathing with its lungs and through its skin. When we do this experiment, we'll use 3 different temperatures of water: very cold water, room temperature water, and quite warm water, no more than 40 degrees C or 100 degrees Fahrenheit. Just to give you an idea how we do this, we'll start with the very cold water by putting a bunch of ice cubes in our tank. When the water gets down to about 5 to 10 degrees C or around 40 degrees Fahrenheit, we want to look at the frog's nostril movements and count the number per minute. Make about 5 trials and average the results, and be sure and record the results. We'll then move the temperature up to room temperature, about 20 degrees C or 68 degrees Fahrenheit, and do the same thing. And finally make it even warmer, up to 40 degrees C or 100 degrees Fahrenheit. Then we'll graph the results. We'll plot the number of nostril movements on the vertical axis from 0 to 35 and the temperature in degrees C on the horizontal axis. You should find that the breathing rate increases with temperature. If the frog dives to the bottom, you won't be able to count the nostril movements because he's now breathing through his skin. In fact, you can hold the frog underwater and get him to switch over to this mode of respiration. Students can also determine their own respiration rates for a comparative value. They can hold their hand under their nose and count the number of times they exhale in 1 minute. Another thing to introduce is the idea that the frog is a cold-blooded animal. And what does this mean? Well, it means that the body temperature of the frog changes with the temperature of the water that it's in. If we could stick a thermometer in the frog's mouth and down into its gullet and measure its temperature in this 10-degree water, we would find that the frog is also about 10 degrees. In fact, this is an activity that's done in the junior high. Let's speculate instead on how the frog's body temperature changes with the water that it's in by putting a graph on the blackboard. The horizontal axis is the water temperature in degrees C, and the vertical axis is the internal body temperature of the frog. Put different lines on your graph and have your students decide which line best represents a cold-blooded animal. The next step is to have them determine what a warm-blooded animal's graph would look like. And of course it'll be a straight line going across at a constant temperature. Students can measure their own body temperature very simply by putting a thermometer inside the corner of their arm and closing their arm on it. After about 2 minutes, they can read the temperature with the thermometer in place. Whether they're inside or outside, the temperature of their body remains constant. It's good that they understand this difference between cold-blooded and warm-blooded. We can do some more animal investigations, this time with a goldfish. But before we do, I should rescue my frog from the ice water. This activity is designed to develop an understanding of and a respect for animals. First, discuss with your class what type of environment is necessary for a goldfish to live in. Is it adapted to live in air? Why not? Can it live in salt water? What about tap water? Can the chlorine harm a fish? And what are some other pollutants that might hurt the fish? After the discussion, break your class up into small groups of 2 or 3. Give each group a plastic cup and a thermometer. Have them prepare a sample of water that the goldfish can live in. If they're using tap water, make sure that they dechlorinate the water with 2 drops of water conditioner. You can get this at a pet store and it works immediately. Students will also use their thermometer to make sure that the water temperature is within a prescribed range. A good range is from 65 to 70 degrees Fahrenheit, or about 20 degrees Celsius. Now, once they bring their sample up to the front of the room and you approve it, they're ready to scoop out a fish from the big tank using a small net. They take their sample with their fish inside back to the station and start making some observations. What are some of the things that you can observe by looking at the fish? Keep it kind of an open-ended assignment, but make sure that they record all of their observations. How does it swim? How does it breathe? Where are the fins located? Perhaps draw a picture of the fish. If you want, you could have them try and measure the number of gill movements or mouth movements per minute, but this can be quite difficult if the fish decides to start swimming around in circles. Once the activity is over, you want to get the fish back to the main container. Since the dip net won't fit in here, you can very simply do this by pouring the fish into the net and then flipping the fish into the main container. As long as we have a small goldfish here, there's another great activity that we can do with a microprojector, and that's observe the circulation of the blood in the tail of a goldfish. First, recruit a student volunteer who's quick at focusing the microprojector. Then get a Petri dish and soak a piece of cheesecloth and lay it on the bottom. Then you're ready to collect your fish. Just as we did before, pour the water into a cup and collect the fish in a net. Place the fish on the cheesecloth so that the tail is exposed on the Petri dish, and then wrap the cheesecloth around the fish. Spread the tail out if necessary with your fingers and put a microscope slide over the tail. You're now ready to put your Petri dish on the stage of the microprojector and get your focusing assistant to come in and help. This is an excellent demonstration introducing the circulatory system and the importance of blood. Students can file by the screen and observe the flow. They should be able to see individual disc-shaped red blood cells flowing through the vessels. The arteries carry blood away from the heart, and the veins return blood back to the heart. The tiniest of blood vessels are called the capillaries. Some are so small that the blood cells must pass in single file. If you look at the tip of the tail, you can see where arterial blood from the heart turns around and begins the return journey. Don't forget to keep your fish wet at all times and return it to the tank within 5 minutes. While we're investigating fish, another interesting activity is to take a look at fish scales, because they have rings like trees have, and you can identify the age of the fish. The best way to get the scales is from a local fish store or from a fish that you caught yourself. Dry the scales out between 2 pieces of paper towel and a book, and if they start to curl up, you can always wet them before you're ready to use them. What we're going to do is look at them under the polarizing filter on the microprojector. Remember that the microprojector has 2 polarizing filters. First, turn the lower turret to the P position. Then swing the second polarizing filter, which is located above the stage, into position. When we observe the fish scales, we will be turning this upper polarizing filter. The refraction of light through the fish scale produces a beautiful effect when viewed through the polarizers. When we look closely at the rings, we see that some stand out from others, dividing the scale into wide bands. Each individual line is a growth ring, but unlike a tree, many of these rings are made in 1 year. The rings in the center of the scale are the oldest, and the outermost ones are the latest growth. In the winter, the activity of the fish slows down and there's very little growth. Here the growth rings are few and very close together. In the summer, when the food is plentiful, growth is rapid, producing many rings that are spread further apart. We can determine both the age of the fish and even the severity of the winters by observing the rings. This fish appears to be 1, 2, 3, 4, 5, 6 years old. Another topic to investigate is the fact that animals give off carbon dioxide as a waste product. We've already worked with bromothiamyl blue. It's an indicator that shows the presence of carbon dioxide. Remember, it turns from blue to green and then to yellow. When it changes from blue to green, it's showing the buildup of carbon dioxide, and then even more, it turns to yellow. We put bromothiamyl blue in a test tube, and in the plant section we put some Elodea plants in it, and we turned it back the other way. This showed that the plants used up the carbon dioxide when it went from yellow back to blue. Here we're going to take 2 apple snails and put them in a test tube of bromothiamyl blue. After about 24 hours, we'll check the color of the solution to see if it's turned green or yellow. We should also have a control so that we can always make a comparison. Make sure that you stopper up both test tubes and that your students can compare the colors at the beginning of the experiment. A day has really passed, and this is what our results look like. The test tube with the snails in it hasn't turned yellow, but it has turned to a nice shade of green. And of course the control sample is still the bromothiamyl blue color. Make sure that you don't have the snails in here for longer than 24 hours because they can suffocate as they use the oxygen up in the water. When we investigated plants, we saw how plants respond to different stimuli. This was called a tropism. There were geotropisms and phototropisms and so on. Now, when we see how animals respond to different stimuli, we don't use the word tropism, but rather the word taxis. So now we have geotaxis and phototaxis. In this activity, we're going to investigate our old friend from the primary grades, the earthworm. And this time we're going to get a little bit more in depth, and we're going to see how the earthworm responds to different stimuli. To get the most mileage from this experiment, conduct a question period the day before you're ready to do it. First, tell your class that we're trying to test an earthworm to see what it likes and what it dislikes. Ask them to list some things that we could test. Make sure that none of them can be harmful to the worm. Simple things like water, sugar water, salt water solutions are all good. How does it react to light? How does it react to touch? Does it like to walk or slither on sandpaper or glass? What does it do when it comes to the edge of a table? Once you list all the questions that you want to answer, have the students propose a way to test that particular taxus, whether it's geotaxis or thermotaxis or phototaxis. The goal here is to get the students to design the experiment with your guidance. It will take some patience, but hopefully you'll have set up about 10 experiments that your class is ready to do the next day. Let's first set up some logistics. We'll have the students work in teams of 3. Each team will get a plate and a worm. You can buy these worms as night crawlers at the local bait and tackle store. Make sure that the students know which end is the head and which end is the tail. Let's have 9 or 10 different stations throughout the room, and student groups will go from one station to the next and follow the rules at that station, test their worm, and then as soon as they're through, wait for you to release them onto the next station so that everybody changes at once, and they each get to do 9 or 10 different experiments with their worm. They should also make sure that there's water nearby so they can rinse their worm off and make it fresh so if it dries out, it won't die. And also a piece of paper towel so they can always constantly wipe their plate out because you're going to be doing their experiments in their plate. Now, what are some of the experiments we can do? First, let's consider phototaxis. How do the worms respond to light? Do they like light or do they dislike it? Well, here's one possible solution. Get a large test tube and cover it with black plastic. Scotch tape it down so that it's completely sealed. And if you look inside, you can see that it's quite dark in there. When the students get to this station, they'll put their worm tail first in the test tube to see if it goes toward the dark or the light, and then head first. See how it responds that way. Another way to test light is to have a little darkened corner of the room and keep your worm on the plate and shine a flashlight on it. Start at the tail end and then move it further and forward toward the head end and see if there's any difference in response. In other words, where are the light sensors located? Are they down near the tail or are they up near the head? When we investigate thigmotaxis, or the response to touch, there's a couple of different activities we can try here too. One is to touch the worm with your finger. Touch it lightly near the tail end and see how it responds, and then touch it up near the head end and see if you get any more of a response. Make sure every time you do an experiment that you record your results. Another one for thigmotaxis is to place the worm near the edge of a table and see what happens when he gets near the edge. And also you can place a worm on a half on and half off, a piece of sandpaper and a piece of plate glass. In this case, we'll see if the worm likes the rough surface or the smooth surface. Each one of these can be a different station. Another good stimuli to test is how worms react to different chemicals. In this case, we'll be real careful that we don't subject the worm to any dangerous chemical. This is called chemotaxis, by the way. What we'll test the worm on are different solutions of water and vinegar and water and salt and so on. Let me show you what happens when we test our worm with a mild saltwater solution. When the students do this, they'll come to the station, take a few drops with the eyedropper of the liquid they're going to test, and spread it out in a line along the plate. And then direct the worm to head toward that solution. Not push it into it, but see what happens when it gets near it. For example, if it's vinegar, the worm may be able to smell it and push away. What happens when it touches a solution? What will happen when it touches this salt water? Well, let's see. You can see that the worm pulls violently away from the salt water. It certainly doesn't like it. This is a good experiment for learning about different types of taxus. It's also good for learning about invertebrates, in this particular case the annelids or the worms. And this worm is called a lumbricus, by the way. That's the genus name. Lumbricus terrestis, I believe. Another thing that's very interesting about worms is the way they reproduce. So it's a good lead-in to talking about sexual reproduction. Reproduction is a topic that elementary students should learn about. And once again, if your school deals with it on a continuum from kindergarten through 6th grade, your students will develop a mature attitude and, more importantly, an understanding of this traditionally neglected topic. First, review what they should already know: the difference between asexual and sexual reproduction. Is the offspring produced by one or two parents? Review fission in the single-cell protists, and take a look once again at vegetative reproduction that happens in some plants. Also review the flower dissection lab and discuss the fact that sperm unites with egg cells in the flower to produce seeds. Now you're ready for the earthworm. It's a very interesting animal. It's called a hermaphrodite. That means that it has both male and female sex organs all in one worm. But it really requires two worms to cross-fertilize each other. Here's how it happens. When worms mate, they line up head to tail. The clitellum, or that smooth section, secretes a slimy belt which holds them together, and sperm is secreted from a small opening at segment number 15. It travels along each worm to the opposite worm and finds its way to a seminal receptacle at segment 9 and 10. This is kind of like a holding tank for future use. At a later date, the clitellum once again secretes a slime ring, and eggs come out of the ovaries through openings in segment 14. The eggs are deposited in the slime ring. The sperm is then released, which now fertilizes the eggs as the ring is pushed forward. Finally, the ring slips from the body and becomes the cocoon in which the new worms develop. The earthworm has one of the most fascinating methods of reproduction, and this should create an interest amongst your students to want to learn more about how animals reproduce. In their studies, they should learn that the sperm cell is very small. It swims with a whip-like tail and greatly outnumbers the egg cells. The egg cell is much larger. It cannot move on its own and usually contains a large amount of yolk that's used for food for the developing embryo. Bird eggs have the most amount of yolk, and mammal eggs lack yolk and are microscopic in size. When the sperm and the egg unite, fertilization takes place. Now there are two types of fertilization: internal and external. Internal fertilization is a characteristic of birds, insects, and mammals. And here, the sperm is deposited inside the oviduct of the female where it fertilizes the egg. External fertilization is a characteristic of many amphibians and fish. First, the female lays many eggs in the water, and then the male swims by the eggs and fertilizes them. Similarly, we also have internal and external development. Internal development is when the young develop within the mother, as with mammals. External development is a characteristic of birds, fish, and insects. Here the young develop from eggs outside of the mother's body. There's one more area to investigate, and that's the amount of care that the young receive from their parents. Survival is dependent on two main factors. Either the parents produce many offspring, and even without care, some of them will survive, or they produce a few offspring and take very good care of them until they grow up. When we look at external development, usually we see a large number of fertilized eggs and then the parents abandon them. This is true for fish, amphibians, and insects. Here, only a small percentage of them survive, and the ones that do must rely on instinct for survival. When we look at birds and mammals, we see only a few eggs, most of which get fertilized and are cared for by one or both parents for some period of time. Reptiles are interesting in that some give live birth while others lay eggs. Alligators may lay up to 50 eggs at once, then guard the nest and protect the young. In humans, usually only one egg is produced at a time, and the young are cared for until they reach the age of 18. Now your goal as a teacher is to encourage open discussion on these topics. One way to do this is to assign an animal to each student in the class and have them report on that animal's method of reproduction. Each animal can be classified into one of three categories. The first category is internal fertilization and internal development. Examples would be a cat or a horse. Category 2 is internal fertilization and external development. This would be a bird or an alligator. And the final category is external fertilization and external development. And animals in this category would be a fish or a frog. Now for an activity that you can't pass up, and that's taking a look at mealworms and looking at how certain animals, like these little worms, go through different stages of development or metamorphosis. We've already talked about reproduction, so that's a good beginning. Now we're going to see how the egg develops into an adult. And in this case, we're going to look at complete metamorphosis. So we go from the egg to the larva to the pupa to the adult. You can buy mealworms at the local pet store or bait and tackle store, or even order them from a science supply house. When you get your mealworms, they should look something like these. This larval stage is the one stage that the mealworm is in the longest. Let's review how this happens. It starts as an egg, and the eggs are very tiny and they're white in color and they're difficult to find. After about 1 to 2 weeks, the eggs hatch. They become the larva. The larva is very tiny at first and then grows to be about this size. As it grows, it molts its skin or sheds its skin several times. After about 4 to 6 months of growth and constantly eating their way through the bran or the oatmeal that they're living in, they go into a quiescent stage, a stage where they don't move around much, but their body goes through great changes. In this pupa stage, the body becomes hard. It changes from its white color first to a reddish brown color and then to black, and it develops over a period of 1 to 3 weeks. During this period, your students can observe these with their magnifiers and record what they see. It's a very interesting transformation. These little critters almost look dead in this stage, but after about 3 weeks, another transformation takes place as they turn to adult. The adult beetles look like the common black beetles, and although they have vestigial wings, they can't fly. They only live for about a month. And during this time, this short period of time, the female can lay up to 500 eggs. Let's see how you can have your own colony of mealworms in your classroom from just one small sample. First, you need a container. A 5-gallon aquarium works good, or a coffee can, or even a margarine tub, depending on the size of the colony you want to develop. Throw some food in there. Mealworms like bran or chick starter mash, but you can even use oatmeal or any other type of grain. And then get your sample mealworms from the pet store and throw them in. Remember that these mealworms are probably about 4 or 5 months old already, so it's not going to take long until they turn into the pupa stage. Be sure and monitor your sample once a week or so, and when they start to turn to the pupa, make sure that your students get an opportunity to look at them so they're seeing that this transition is really taking place and you're not pulling the worms out one night and sticking some new animal in there to fake them out. It's really happening. There's a metamorphosis going on. Make sure that they see 3 weeks later the beetles appearing. The beetles, of course, will mate and produce tons and tons of eggs, but now you're going to have to wait a lot longer, because 2 weeks until the eggs hatch, and then 6 months as these tiny little mealworms start very small and then grow to full size. There are a few more things you should know before you develop your colony. First, don't let them live in cardboard or paper containers, because they can eat their way out. And if you have a tight-fitting lid on your container, be sure and punch a few holes in it, because they are animals and require oxygen. They don't require much moisture though. In fact, they can get most of the moisture that they need from the food that they eat. They have an interesting adaptation that they conserve almost all of their moisture, and their waste product is in the form of a dry pellet. However, once you get to the beetle stage, they actually sometimes will cannibalize the pupa and eat them for the moisture, because the beetles need more moisture. In this case, it's good to throw in a small slice of apple or carrot or potato to help them get the moisture that they need. You don't need much though. Also, it's a good practice to change the growing material, the bran or the mash starter, whatever you're using, on a 6-month interval, and then clean out all the molted skin that might be mixed in with it. And don't hesitate to let this developing colony sit in your classroom over the summer while you're gone away on vacation. When you teach a science lesson, it's almost inevitable that sometime you're going to get some unpredictable results, and before you know it, you're on a whole new lesson. You've gone on a tangent off from your original plan. It's important to be flexible here and allow you and your students to go off and learn new things. Let me give you an example. I was preparing a segment for the ecosystem section of this tape on the food chain, how one animal eats another. And in this case, I was working with a microscopic animal called a hydra. It's related to the jellyfish and the sea anemones, and it has tentacles. And I was feeding it a Daphnia. This is interesting to watch because you can see how the hydra is adapted to capture prey with its tentacles and also observe the food chain in action. After the Daphnia was captured, I noticed something inside was still moving. It appeared to be 2 young embryos ready to hatch. Now I had read that you can lightly press down on the animal's body with a pin and stimulate birth. So why not try it? With a steady hand and lots of luck, out popped a young Daphnia in the larval stage. Absolutely amazing. As a result of this deviation, my lesson now fit better in this section on life cycles. Incidentally, we rescued the little fellow with an eyedropper and transferred him back to the main jar. The phylum of arthropods is the most numerous and widespread group of animals in the world. The word arthropod means jointed leg and includes subdivisions or classes of insects, crustaceans, spiders, and centipedes. The mealworm, incidentally, is not a worm at all, but actually an insect. And the water flea, or Daphnia, which we just looked at, is in the class of crustaceans. We're now going to take a look at another crustacean, the saltwater relative of the Daphnia. It's called the brine shrimp, or the Artemia salina, or fondly known as sea monkeys. And once again, we're going to involve our students as much as possible in the design of this experiment. First, we need to make a solution for hatching the eggs that's similar to that of seawater, about 3.5% salt. How do we make this 3.5% solution? Well, remember there's 96.5 milliliters of water plus 3.5 grams of salt to bring the total weight to 100 grams and give us the proper concentration. And if you don't have the balances or graduated cylinders, you can always fake it with 1/2 cup of water and 1/2 teaspoon of salt. You might talk to your students why we decide to use distilled water instead of tap water because of the chlorine, and why we're using regular salt instead of iodized salt. Once you've mixed the solution up, it's time to throw in a pinch of brine shrimp eggs and wait about 48 hours. During this time, students can take eyedroppers and suck up a small sample and take a look at it with a magnifier. This is what it should look like after 2 days have passed. If you have a microprojector, you'll get a much better view of what these little larval forms look like. Also during this 2-day period, we'll be designing some experiments. And once again, get the students involved and let them decide what they want to test. We're talking about taxis again. This time, how do brine shrimp respond to different stimuli? I'm using microscope slides with clay barriers placed on them, and I'm putting the brine shrimp in the middle. We're going to test them to different stimuli. Let's try phototaxis for an example. I'll put a piece of black paper over this end of the barrier. Will the brine shrimp swim toward it or away from it? And let's not forget the control, the unaffected sample sitting next to the experiment to make sure that it's really the light that's making them move that way and not some other factor. We can also test chemotaxis. In this case, we'll use a piece of thread and we'll test different chemicals. Let's say we want to test vinegar. Here's some vinegar in here. We'll dip the thread in the vinegar, get it so it's soaked, and then lay it across the experiment sample. You should also lay a piece of thread across the control, but this one's not dipped in vinegar. We want to make sure that the brine shrimp aren't reacting to the thread, but rather to the vinegar. What about thermotaxis? How would a student design that type of experiment? Well, we could set half of our slide on ice and the other half on a piece of wood. Let's take the thread out of here and set this up like this, with a piece of ice right here. Now if the brine shrimp like cold, they'll swim toward this side of the slide. If they don't like the cold, they'll move away from it. After about 3 or 4 minutes, we can look at our slide and see which way they go. Once again, we should probably have our control sample sitting nearby to make sure that there's not some other external influence that's making them move in a certain direction. We also have the reaction toward electricity. This one's kind of fun. We'll take a battery, and in this case, we'll get our slide back here, and we'll put the electrodes the plus on one side and the minus on the other, inside with the brine shrimp and see how they react. Will they swim toward the plus electrode, the minus electrode, or not respond at all? Perhaps a student would like to see how brine shrimp react to magnets. Nothing wrong with that. Hold a magnet near one end of the slide and see if they swim toward the magnet. And don't forget your control. When we study geotaxis, whether they go up or down, it's pretty difficult with a microscope slide because we can't turn one on its end. You might try this with a couple of test tubes, maybe as a demonstration, with one sample upright and one sample on its side to see if they go any particular way. Students can record their results on a lab sheet. You can put before and after pictures and they can place dots in to represent the location of the brine shrimp. Remember that there is no right or wrong answer in each experiment. The results of each particular group is what they should believe in. You can get the consensus from all the groups by putting the data on the blackboard and looking at the most probable response of the brine shrimp to different stimuli. The most important part of this whole experiment is not finding out what brine shrimp do, but getting the students involved in the experiment process, designing their own experiments, controlling variables, and learning how to solve problems. Now for a few miscellaneous ideas before we go on to the next section. First, you may want to have a spare cage in your classroom for that unexpected visitor. This one's made out of hardware cloth and wood, and it's quite inexpensive. It even has a little door that opens up on a hinge. The next one is made out of a fish tank, so if you have a spare fish tank, it's just a matter of folding up some hardware cloth to fit over the top. You can also make your own complete cage out of hardware cloth by making a cylinder and then connecting it with a piece of baling wire. And then it's a matter of get a few cake pans and just slide it right over the bottom and the top, and you have an instant animal cage. You can make smaller ones too for insects out of tuna cans, and it would look something like this. Notice that this wire mesh has smaller holes. Another thing you might like to try is attracting birds to the vicinity of your classroom. You can make some very inexpensive bird feeders out of half-gallon milk cartons and hang them from a tree near your room. You can fill them with wild bird seed that's available from grocery stores. Another fun activity to do is to make plaster casts of animal footprints. Here's how to demonstrate it. First, get a cookie tin and fill it with mud. Find your local neighborhood cat, and when the mud is just the right consistency, press the foot into the mud and then let it dry for about a day. After it's somewhat dry, bring it into class and show it to your students. If the mud is loose enough, you can use a paper collar and press it into the mud. If it's dried hard, you may have to use a piece of clay like we did with the brine shrimp to make a seal around it. Then mix up the plaster, 2 parts plaster and 1 part water, pour it in and let it set for about 30 minutes. After 30 minutes, you can peel it off and show it to your students. Then give them the opportunity to look for animal footprints around their home. Give each student a sample of plaster and let them take it home and make a cast of the footprints that they find. Have them bring the casts in and show them to the rest of the class, and perhaps even write a short story about the animal who made that footprint. Too often students have misconceptions about animals. For example, by watching Saturday morning cartoons, they start to see the coyote as the bad guy and the roadrunner as the good guy. But it's important to dispel these misconceptions. By understanding and learning about animals, we develop a respect for animals, and that's very important. Let's now go on and look at one more animal, the human being.